Welcome to this course on metabolic regulation. Metabolism is the totality of chemical reactions that occur in living organisms to maintain life. These processes are tightly regulated to ensure the appropriate allocation of energy and resources, depending on the physiological needs of the cell or organism. Metabolic regulation refers to the systems and mechanisms that control these chemical processes. It ensures that enzymes, substrates, and pathways operate at optimal rates, and it integrates internal and external signals, such as hormonal fluctuations and nutrient availability. This course aims to introduce hormonal and molecular mechanisms of metabolic control, explore how metabolic pathways interconnect and interact, examine enzyme regulation mechanisms such as feedback inhibition, catabolite repression, and operant control systems. Understand the role of compartmentation, turnover rates, and metabolic pools. Let us begin by exploring the central control center of metabolism, the endocrine system. Lecture 2: The endocrine system and its role in metabolic regulation. The endocrine system is a complex network of glands and organs that produce, store, and secrete hormones, chemical messengers that regulate metabolism, growth, and homeostasis. 1. Major endocrine glands. Pituitary gland, the master gland that releases tropic hormones to control other endocrine glands. Thyroid gland, produces thyroxine and triiodothyronine, increasing basal metabolic rate. Pancreas, secretes insulin and glucagon, major regulators of blood glucose. Adrenal glands, release cortisol and adrenaline, involved in stress response and energy metabolism. Gonads, secrete sex hormones, estrogen, testosterone, which influence metabolic rate and energy storage. 2. Hormonal control of metabolism. Hormones act through receptors, triggering intracellular signaling cascades that regulate enzyme activity, e.g., activation/inhibition. Gene expression, e.g., transcription of metabolic enzymes. Transport proteins, e.g., glucose transporters. 3. Examples of hormonal regulation. Insulin promotes glucose uptake and storage as glycogen and fat. Glucagon stimulates glycogenolysis and gluconeogenesis. Cortisol increases protein catabolism and gluconeogenesis. Epinephrine triggers rapid glycogen breakdown during stress. Hormonal imbalances can lead to metabolic disorders such as diabetes, hypothyroidism, or obesity. Lecture 3: Conditions, Classes, and Experimental Approaches to Metabolic Control. Understanding how metabolism is controlled requires a grasp of the conditions under which regulation occurs, the types of regulatory mechanisms, and the methods used to study them. 1. Conditions of metabolic control. Nutrient availability, fed versus fasting state. Energy demand, resting versus exercising. Hormonal signals, insulin versus glucagon. Developmental stage, growth versus aging. Stress response, inflammation, trauma, infection. 2. Classes of metabolic control. Allosteric regulation, enzyme activity is modified by binding of small molecules. Covalent modification, phosphorylation/dephosphorylation can activate or deactivate enzymes. Transcriptional regulation, changes in gene expression control enzyme synthesis. Substrate availability, concentration of substrates can drive reactions forward or backward. Hormonal signaling, often linked to second messenger systems like CMP, IP3, or Ca superscript 2. 3. Experimental approaches. Isotope tracing, e.g., C13, N15 track metabolic flux through pathways. Enzyme kinetics, measure Vmax and Km to evaluate enzyme efficiency. Western blotting, quantify protein levels and modifications. qPCR and RNA sequencing, analyze gene expression profiles. Metabolomics, comprehensive profiling of metabolites using LC-MS or NMR. Gene knockout/CRISPR, study the effect of removing specific metabolic genes. These approaches help unravel how cells maintain energy balance and respond to changes in their environment. Lecture 4: Integration of Metabolic Pathways. Metabolism is not a set of isolated pathways, it's an interconnected network that shares intermediates and regulatory signals. This integration is essential for coordinated energy flow. 1. The Krebs cycle, citric acid cycle, as the hub. The Krebs cycle lies at the center of metabolism, linking carbohydrate metabolism, through pyruvate from glycolysis. Fat metabolism, through acid-CoA from beta oxidation. Protein metabolism, through deaminated amino acids that feed into cycle intermediates. Nucleotide metabolism, supplies intermediates like fumarate and succinyl-CoA. 2. Examples of interconnection. Glucose left-right-arrow-pyruvate left-right-arrow-acid-CoA left-right-arrow-citrate. Amino acids left-right-arrow-alpha-ketoglutarate, oxaloacetate, fumarate. Fatty acids left-right-arrow-acid-CoA left-right-arrow-ketone bodies. 3. Reciprocal regulation. Glycolysis versus gluconeogenesis, regulated by ATP, AMP, fructose, 2,6-bisphosphate. Glycogenesis versus glycogenolysis, controlled by insulin and glucagon. Lipogenesis versus lipolysis, driven by energy status and hormonal balance. Cells integrate signals from multiple pathways to maintain homeostasis and respond to energy needs. Lecture 5: Turnover rates and metabolic pools. Metabolic regulation also involves flux control, turnover rates, and the concept of metabolic pools. 1. Turnover rate. This refers to how quickly a molecule or pathway is synthesized and degraded. High turnover equals rapid response, e.g., ATP synthesis. Low turnover equals long-term storage, e.g., triglycerides in adipose tissue. Turnover ensures that damaged or outdated molecules are replaced and enables dynamic control. 2. Metabolic pools. Metabolites are grouped into functional pools. Free amino acid pool supplies substrates for protein synthesis and energy. Hexose phosphate pool, glucose 6-phosphate, fructose 6-phosphate, used in glycolysis, PPP. Acid-CoA pool, common intermediate in carbohydrate, fat, and protein metabolism. Maintaining appropriate levels in each pool is crucial for metabolic flexibility. 3. Isotopic studies and flux analysis. Using labeled compounds helps measure the turnover of ATP, glucose, lipids, and more. This sheds light on metabolic priorities under various physiological conditions. Lecture 6: Significance of compartmentation in metabolism. Eukaryotic cells use compartmentation to separate and coordinate metabolic activities. 1. Subcellular compartments. Cytosol, glycolysis, pentose phosphate pathway, fatty acid synthesis. Mitochondria, Krebs cycle, beta oxidation, oxidative phosphorylation. Endoplasmic reticulum, lipid synthesis, detoxification. Nucleus, DNA/RNA metabolism. Peroxisomes, very long-chain fatty acid oxidation. 2. Functional advantages. Prevents interference between incompatible reactions. Enables localized control and regulation. Creates concentration gradients. Allows for differential enzyme expression. 3. Examples. Acid-CoA produced in mitochondria but needed in cytosol for lipid synthesis. NADPH produced in cytosol, PPP, but used in fatty acid synthesis. Transporters and shuttle systems, e.g., malate aspartate shuttle, facilitate cross-compartment communication. Module 1 Summary. In this module, we covered the role of the endocrine system in metabolic regulation, the conditions and classes of metabolic control, key experimental methods used to study metabolic regulation, integration of metabolic pathways around the Krebs cycle, the importance of turnover rates and metabolic pools, the functional significance of compartmentation in metabolism.